Hello and welcome to the Anitox IPP Tech Talk on supporting advances in salmonella mapping in poultry production with CRISPR sequencing. My name is Jose Ramirez and I'm a technical director for Anitox. I am delighted to be your host for this session. Clearly, we would rather be able to meet face to face at the IPP this year, but sadly, that's not the case. So instead, we will take this opportunity to present to you and welcome you to reach out and follow up online after the session. But more about that later. In 2018, Anitox signed up to support a research program to establish whether CRISPR technology could effectively map the relationship between safety-critical salmonella in feed and poultry products. We were delighted to be able to support the work, which is being undertaken by Dr. Nikki Sherrod, Assistant Professor at the Department of Population Health, Poultry Diagnostic, and Research Center of the Center for Veterinary Medicine at the University of Georgia. Since the time of recording, Dr. Sherrod and her team have published the work. Again, more about that shortly. But first, I caught up with Dr. Kurt Richardson, Anitox Chief Science Officer Emeritus, and a microbiologist of some 40 years standing. Dr. Richardson has himself published multiple papers on salmonella analytical techniques. Dr. Richardson and Anitox laboratories have been preparing hundreds of feed samples for Dr. Sherrod over the past 18 months. I asked him to explain why Dr. Sherrod's work is proving so fundamental to our changing understanding of salmonella serotypes at various points in the poultry processing. Dr. Richardson, over to you.
CRISPR is a technology that Anitox learned about with respect to salmonella diagnostics about 2 years ago, and we got very excited when we saw what it was capable of doing. The researcher that we visited was up in Gettysburg, Pennsylvania, and has relocated to PDRC, so it's a very good relationship. We're close, we can keep up, learn new technologies, and what's going on in the world of CRISPR. In today's world, there's a lot of pressure on the producer to control salmonella and other foodborne pathogens in their products that they deliver to the consumers. In the United States, the USDA sets limits for salmonella in broilers, turkeys, and swine. They're also talking about setting limits for beef as well. In today's world, 15% of our processing plants, poultry processing plants, are actually failing the USDA standards, and their names are published on a monthly basis of who passes and who fails. The FDA also regulates food safety with respect to eggs. Right now, the regulation is specified for Salmonella enteritidis, but they have expanded it to other salmonella serotypes. So there's a lot of regulatory risk, but there's also risk from the consumer. One of the challenges at protecting your brand name or complying with FDA and USDA regulations is, so you've got a salmonella problem. Where did it come from? Where do you put your money as far as solving this issue? At the current time in the United States, we put all of our money in our processing plants. We spend a lot of money treating carcasses with chemicals to reduce salmonella risk, but plants are still failing. And what that means is we have to go back upstream. Well, the isolates that we find in the processing plants don't match the isolates we find in at the farm. We can't find the isolates in the breeders, in the feed, in the environment. And it poses a problem. You know, we go out in the field and our customers will tell us in the United States that the isolates they find in their processing plant don't correlate to the isolates in their feed, and that feed is not a vector in their mind for salmonella. Next slide, please. But there's a problem with this assumption. You know, when we look at salmonella by the traditional methods, what we do is we collect a sample. We do a pre-enrichment, and from that enrichment, pre-enrichment, we plate it on agar, and we pick 1 to 2 colonies of salmonella off that agar plate and we serotype it. And we say whatever we find in those 1 to 2 colonies off of that tremendous field of salmonella is what is there. And that's not necessarily the case. Next slide, please. Picking 1 to 2 colonies selects the dominant serotype, what grows the best in the media, what grows the best on the agar plate. There's other serotypes that are on that plate that we don't see, that are in lower populations. And in a lot of cases, those minor serotypes are the ones that are important to food safety. In some of the preliminary work we saw by PDRC on processed chicken in the United States, they found Salmonella Kentucky as the predominant serotype. And they found small levels of Salmonella enteritis and Salmonella typhimurium, which are the #1 and #2 serotype of Salmonella causing human food disease in that sample. And CRISPR is a tool that can help us identify those minor serotypes. So we'll go into a little bit about what CRISPR actually is. Next slide, please. CRISPR is an acronym. Nobody can pronounce clustered, regularly interspaced, short palindromic repeats twice in a row. So we use the abbreviation or the acronym CRISPR to describe it. And what a palindromic repeat is it's like the word madame. If we spell it forward, it says madame. If we spell it backwards, it spells madame. Same with radar. In the case of CRISPR, the individual nucleotides that make up DNA, for example, AAC and CAA, are acronyms. Now, CRISPR sequences are only found in bacteria. 50% of the bacterial populations have CRISPR repeats. And what these repeats are is it's genetic information that viruses have inserted into the DNA of the bacteria. And it's passed along from generation to generation. Next slide, please. Now, how CRISPR works is there's an enzyme called Cas9. A lot of times when people refer to CRISPR, they'll say CRISPR Cas9. Now, this enzyme recognizes these repeats in the genetic information, and it cleaves those sequences out so we can isolate and identify them by PCR. Next slide. Now, in the case of Salmonella, we're very fortunate. Salmonella has 2 CRISPR locations or 2 CRISPR loci. And what that allows us to do is it allows us to structure the 2,500 serotypes into individual patterns. You can see on underneath the CRISPR1 loci, the genetic information of the Salmonella. And you can see in those high cap numbers, uppercase numbers, that's the repeat segment that we're looking for. So what the enzyme does is it cleaves out that repeat and breaks it down into a CRISPR profile. Go ahead. Next slide. And then we do this CRISPR array. We associate these repeated sequences into a pictorial, as we can say. And each Salmonella cerevar has a different profile. So we can differentiate between our Salmonella serotypes very easy. Now, in the lab, when we're doing Salmonella serotyping, we're doing antibody testing. We're testing what serogroup it belongs to, and then we have another step and then another step trying to distinguish which Salmonella we have. With CRISPR, it's all in one. You don't have to go through multiple steps, and it's less expensive. Next, please. But the real advantage of CRISPR is it's not relying on an individual colony that we select off the agar plate. It's relying on the population of Salmonella. So what we were interested in is not identifying the Salmonella strain. We can do that by immunoassay. We can do that by whole genome sequencing. We want to know if we mix multiple serotypes and we extract that DNA, can we see all the serotypes of salmonella that are in that sample and get away from picking that 1 to 2 colonies off that plate? Go ahead. Next slide. So what we set up to do with Anitox is to validate the CRISPR to the traditional methodology for animal feeds. This is the first step. If I do a traditional method and this is what I get, will CRISPR give me the same answer? The second thing is to determine the limitations of CRISPR. You know, how sensitive is this? If I have Salmonella enteritis in a sample and it's in one part and I have 100 parts of Kentucky, can I find that enteritis? Well, what if I had 1,000 parts of Kentucky? Can I still find enteritis? What about 10,000? What about 100,000? And then to take this technology once it's been validated and look at animal feed. In all the surveys that we look at throughout the world, the data is actually flawed with respect to salmonella in feed and feed ingredients. Researchers are picking 1 to 2 colonies off a plate, and they're saying that's what's in the feed sample. But if you look at some data from back in the 1950s, they showed that feed could have as many as 9 different serotypes of salmonella. And we base all of our risk assessment on the one that we select. So we want to understand how we can use CRISPR for that. And then to conduct biomapping, to work with an integrated broiler producer or integrated turkey producer, to trace the different salmonella serotypes through their operation and determine where the real risk is. Is it the baby chick or is it the feed? What role does the feed play in carcass contamination, what they see at the processing plant? Next, please. So step 1 is underway. We're looking at CRISPR versus the traditional method, and we have some preliminary data that have come in that I'll share with you. But what we did in our laboratory is we mixed 2 to 4 different strains of salmonella together. We mixed 2 together, we mixed 3 together, and then we mixed all 4 together in equal proportions. And then we did a pre-enrichment. And from that pre-enrichment, we took 50 colonies off an agar plate. You know, most people take 1. We took 50. We wanted to know what in that plate, if 1 to 1, would we get 25 and 25, or would we get 90 and 10? And then we took those samples that we had plated and we sent them off for CRISPR analysis. We did the same with enrichment medias, and we used 2 medias in this case. Next, please. Next slide. Well, what we've learned is not surprising to us. It was surprising to our partners up at USDA and PDRC, is that when we mix populations together in 50-50, only in 2 instances did we get 50-50 back. Some serotypes outgrow or outcompete other serotypes in pre-enrichment media, which is the universal pre-enrichment broth. For example, when we mix Seffenberg and Montevideo together, we got 90% Seffenberg and only 10% Montevideo. We didn't get that 50-50 mix. And when we first started getting preliminary data back from PDRC, they couldn't understand this either, but it's normal. We have 2,500 serotypes, and we have a generic media. And all 2,500 don't grow the same. That should be common sense to me, but others don't understand that. When we take that universal pre-enrichment broth and we put it into something like an enrichment broth. Now the RV stands for Rappaport Phaceolus, which is the enrichment broth used in Europe and used in the United States. And the TT stands for tetrathionate broth. When we take it to the next step, from the pre-enrichment to the enrichment, we see that the ratios of these serotypes change. For example, in our universal pre-enrichment broth, when we mixed typhimurium and Montevideo together, we had a 50-50 mix. But when we put it in RV broth, it was 25% typhimurium and 75% Montevideo. So we are biasing our serotypes as we move through our pre-enrichment to our enrichment broths. And we're influencing what we see at the end, what that 1 or 2 colonies that we see by the traditional method. Now, CRISPR can differentiate between this. It was interesting when we were doing this, we had an isolate of Tennessee that we were working with, and the researcher came back and she told us, well, your naturally contaminated material contains 0.1% enteritis. We plated this media multiple times and never saw that. But with CRISPR, she was able to see it. So we had to take some new steps in the lab to prevent potential cross-contamination in our inoculums. Next, please. We're not alone in our goal of using CRISPR as a diagnostic tool. There's a recent study that was published in December 2019 by Nelson Cox and PDRC that looked at broiler carcass samples. They saw the exact same thing that we saw. Pre-enrichment and enrichment media influences salmonella serotypes that we detect. So the traditional method that we're using won't pick up those minor serotypes that we're looking for. We'll only find the dominant serotype. They also reported that CRISPR and the traditional methods have an 86% correlation with respect to the predominant serotype, the major one. And what that shows to us is that traditional methodology is flawed. 14% of the time, the predominant serotype is not the one we selected off the agar plate. We were selecting something else. So we get a mixed picture. And you can see when we get this mixed picture of why the poultry industry is confused about where risk really is assigned. You know, is it the feed? Is it the farm? Is it the hatchery? Is it the breeder? All they know is this is what we see, and we have no idea where it comes from. They also did what we're working on, stage 2 of our research program, is what sort of limitations does CRISPR have? How sensitive is it? And what they found is that they could detect one salmonella serotype out of 20,000 salmonella colonies. So their sensitivity is extremely low, or high, I would say. What we're looking for in feed, the minor serotypes that we're looking for, there's a good success of finding those in a population that is predominantly a serotype of, we'll say, non-foodborne disease. They also saw what we're hoping to see in our feed study that is underway right now, is that carcasses had between 4 to 9 different serotypes on them. And when we talk to a processing plant, they say, I only find Kentucky on my carcass. But what this data coming out of USDA and PDRC says, well, you find Kentucky, there's 8 others you're not finding. And those are the ones that we have challenges with.
Thank you, Dr. Richardson. As I mentioned earlier, since we recorded that with Dr. Kurt, Dr. Shirad has published the findings of her work with feed sample from the Anitox lab in the Journal of Applied Microbiology. You can find the link here or at our website at www.anitox.com. What we learned is that 7 serotypes of Salmonella were isolated from feed samples, with Salmonella infantis being the predominant one. Salmonella infantis, I'm sure you all recall, was associated with food safety issues in the poultry meat in the years 2019 and 2020. Salmonella Heidelberg was the second most common isolated. This serotype is responsible for the Foster Farms broiler meat recall. In addition, other serovars have been isolated, including Salmonella reading, associated with the 2019 turkey meat recall. Salmonella typhimurium, the second most common cause of salmonella in humans, was also isolated. In addition, Salmonella branderup, the serotype that forced Rose Acres to recall eggs some years ago. Of the 7 serovars isolated, 5 have been associated with human illness. As analytics tools develop, the better we will be able to understand the critical importance of feed as a fomite for food safety and the performance limiting conditions in poultry. Fortunately, there are proven and effective mitigation strategies that we can put in place. To find out more about Terminate, Finio, and the power of clean feed, please visit the Anitox virtual IPP booth at www.anitox.com or reach out to your local Anitox representative. Meantime, thanks for joining us.